And thanks to Tom and thank Kathy and to the Paul Allen Foundation for funding the research I'm going to present today. This is a collaboration between myself and Rachel Wong and Fred Rieke. And it's been a very productive collaboration so far. And we're about a year and a bit into this project. And so I'm going to give you an update progress report. So the basic goal was motivated by the fact that a lot of neural diseases require or are problems of synaptogenesis. And we've heard a little bit about autism, and some of the genes that are related to autistic disorders are genes that are involved in synaptogenesis. Synaptogenesis is a relatively late part of the neural maturation process during development. Synaptogenesis and axon guidance usually occur after the neurons are generated. And in many of the organoid cultures that have been developed so far, synapses are made, but they're just sort of at the beginning of being made in these cultures. And in general, in cells derived from embryonic stem cells, neural cells derived from embryonic stem cells, the process of making synapses and the formation of circuits is really not as well developed as one would like. So we thought, well, perhaps we could study this in the retinas, where the characteristics of the circuits are really well described so far. And by understanding just how well does a retinal circuit develop, we might end up with principles that could allow us to make maturations of neural circuits more general. In addition, I'll describe work that shows that microRNAs are involved in the neural maturation process in the retina in setting this developmental clock. And we thought to apply those, some of the same techniques that we had already studied in normal developing organisms to iPS cells. So I'll just start by jumping right in and telling you a little bit about the retina. The retina is a sheet of neural tissue at the back of your eye that transduces the light responses, the changes in light to changes in electrical potential. And those are relayed, let's see, do I have a point two? Those are relayed from the photoreceptors that first respond to light through bipolar cells that are sitting in the inner retina, and then finally to ganglion cells that coalesce in the optic nerve that go to the brain to transmit the pathway. These cells are organized in three layers. There's the outer nuclear layer, which contains the photoreceptors, inner nuclear layer, which contains the bipolar cells and amacrine cell population, and then a ganglion cell layer that sends the projection neurons to the brain. In between these layers of cells are the synaptic layers. And so that's one of the nice things about the retina is we can identify the formation of the synaptic layers just by looking at the tissues, sectioning it, and seeing that these layers form appropriately. In addition, there's lots known about these various cell types. I'm giving you a very brief overview, but we have details about various types of bipolars, various types of amacrine cells, and ganglion cells, and photoreceptors. And so we can even ask questions about how well do the connections form, how accurately do they form, and ultimately, how well do they approximate in vivo developing retina. So the retina initially forms as an outpocketing from the neural tube during development. So you can see an optic vesicle forms, and the optic vesicle then develops into the optic cup. And then once an optic cup forms in the embryo, there's a set of progenitor cells within there, these multipotent progenitor cells that give rise to all the different types of retinal neurons. I've just highlighted if a single one of these progenitor cells is labeled, it gives rise to a diverse set of retinal cell types. And so basically the way we understand this system is that there's a progenitor cell that's multipotent and can, on its last division, make any of a number of different retinal cell types. Now, over time, the retina is built up from early cells that are generated, like the ganglion cells, cone photoreceptors, and horizontal cells. And then these progenitor cells continue to cycle, and they add new neurons until finally the last neurons that are added into this circuit are the bipolar cells. And the bipolar cells then mark the kind of end of the normal retinal neurogenesis pathway. And these are the cells that we're actually focused on as part of this project, because as they're the last ones to fit into the circuit, they also transmit the circuit information from the photoreceptors here into the ganglion cells. Now, these retinal neurons are generated over time during development. So there's an early cohort, which I've said are the cones, horizontals, and ganglion cells. And then there's a late cohort, the rods, the bipolars, and the mular glia. And like elsewhere in the nervous system, the glia are among the last cell types generated in the central nervous system. This sequence is conserved across all vertebrates, from zebrafish all the way up to macaques and into humans. And so again, you see ganglion cells, horizontal cells, and cones are generated early, followed by this second cohort. And this sequence is similar to the sequence of neural generation that occurs in the cerebral cortex and other areas in the central nervous system, where you have different layered neurons in the cortex generated at different times during development. So the fact that neuroprogenitor cells follow sort of a clock where they produce different cell types at different stages in development, it's a very robust phenomenon. It's been described since people first did thymidine birth dating in the nervous system, and it's not really well understood, even after all the many years of appreciating this clock. However, there's this clock, as I've said, this developmental timing runs in all vertebrates, but it's really quite different in terms of the time span over which it occurs. Obviously, we know a mouse develops and is born after 3 weeks of gestation, and humans after 9 months. So there's a huge difference in species-specific developmental time, and that's reflected in the retina as well. So in mice, you have the period of neurogenesis and development stretching from E15, or really E12, up to about postnatal day 5, and then synaptogenesis starts soon after that. And so we can follow this process in a normal mouse. But in humans, this process takes place starting at around fetal week 7 and then continues. The outer nuclear layer synaptogenesis first starts forming here at about fetal week 13 or 14. And so we know some of the things that describe both the mouse and the human, but the circuitry develops with these same stages in both species. So we're interested in both trying to establish this or understand this process in both mice and in humans. And so we're using iPS or ES cells derived from both human and mouse to study the process in organoids. Now, we first developed a protocol many years ago for growing cells from human ESCs, and we've in our lab and other labs have found ways to grow similar kinds of retinal organoids from mouse. In this case, we have a RAX GFP reporter mouse that allows us to grow organoids and track these organoid formation from ES cells. And it forms similarly to what you see in vivo. And what's interesting is the GFP in these RAX GFP mouse ES cell-derived organoids turns on at around day 7, very close to when it turns on in vivo. So the clock of development in organoids is about the same. It's almost within a day the same as it is in vivo. So this was a phenomenon that first attracted my attention because in humans, in human retinal cultures, we had found that if we take ES cells and make organoids from humans' retina, they take about 3 months to get the first evidence of rhodopsin expression, of rod formation. And in mouse ES cells, it takes about 3 weeks. So this got me wondering, why would there be such a difference between species? The numbers of cells that are produced in these organoids is vastly smaller than what happens in the normal retina, particularly in the human. So they're not counting cell divisions. And they don't seem to be, it's clearly these things are developing outside of the context of the rest of the embryo. We pretty much get the whole embryoid body to turn into a retina. So it's not sort of taking its cues from some other part of the animal. There's not like a coordinated clock across the animal. So we're very interested in trying to understand what regulates the time of development in these neural structures, and how does that relate ultimately to timing in an organism, and then to try and figure out ways of speeding that process up. So that's our overall goal. This just shows you the nice differentiation we can get in these organoid cultures in the retina. Here's the 3 layers that I told you about earlier that form normally in vivo. Well, this is an ES cell-derived retina. Photoreceptors here, internuclear layer cells here, the bipolars, and then the ganglion cells. And some of the photoreceptors develop very nice morphology. They look very similar to what photoreceptors do make in vivo, and they start expressing markers of synapses. And in the retina, we have very specific markers of a synapse type called the ribbon synapse, and we can even see evidence of ribbon synapses forming the organoids between the layers. So at least the mouse cultures are developing very nicely up to the stage of initial synapse formation. And so that's what we're further characterizing now in our system. The human organoids also develop very nicely, but as I said, they're very small. And this is work taken from another group, but they nicely shows the timing recapitulates that of the normal human. And so, for example, at 5 weeks, you start getting ganglion cells and cone photoreceptors, but it's not until 21 weeks that you start seeing evidence for bipolar cells and for rod photoreceptors. So again, this clock ticks even though these things are much smaller than normal. Now, in trying to understand time and biological time, I just want to give you a little bit of history in terms of how this has been talked about over the years. Haeckel first identified heterochrony as or really invented this term as a way to point out that there were changes in the relative time of developmental processes between ancestors and dependents. And he actually viewed this as a departure from his biogenic law that ontogeny recapitulates phylogeny, because these were things that were sort of out of normal time. De Beer really elaborated how heterochrony could actually, differences in timing of some of these processes in development could actually create large amounts of diversity among organisms. You just keep one developmental process on longer, and you can really change the phenotype of that animal relative to an ancestor. Gould really reviewed this area and emphasized that size and differentiation and sexual maturation could be independent variables in terms of these clocks. But there was a refocus, and sorry for this very brief history, but there was a refocus more recently, Rudolf Ralph and Ambrose and Horvitz, on some genetic changes that, actually trying to identify genes that change the timing of developmental processes. And I think this paper by Ambrose and Horvitz is really quite remarkable in that they first identified a sort of a heterochronic pathway. And what this is that they found that in mutants of the nematode C. elegans, that mutations in genes called LET7 and Lin28, LET7 a microRNA and Lin28 a RNA binding protein that regulates this, that these mutations could affect the timing of events during development, such that in LET7 you might have a particular developmental process repeated over and over. Lin28 mutants that negatively regulate this caused a truncation of normal development. And so this heterochronic pathway has been really one of the mainstays of the field as showing how this kinds of processes could work. Well, I saw that paper years ago and thought, oh, that's very interesting. Maybe that's how the timing and development of the nervous system works. But then I really didn't do anything about it and just noticed that. But 10 years went by, and a student in my lab actually did a dicer knockout for a completely different reason. And when we conditionally knock dicer out of the retina, it gets rid of all the microRNAs. And when he did that, essentially the clock stopped at the early stage. So this red marker is ganglion cells, and the retina just keeps producing the ganglion cells over and over and over again. And it never progresses to where it makes rhodopsin. These gaps show the deleted region with no dicer, and it never makes glial markers. So the clock basically stops when you knock out microRNAs. So we went and looked to see what microRNAs were involved in this, and we found indeed that LET7 and Lin28 were part of this pathway. I don't really have time to go through all the data, but this is all published. But basically what we find is that in the mouse retina, a dicer knockout just keeps preserving this early stage, and the cells just keep dividing and making that cell class. Whereas knockouts of Lin28 or overexpression of Lin28 gives the same phenotype with a dicer knockout. And you can rescue the dicer knockout with LET7, miR9, and miR125, parts of the C. elegans pathway. So our idea was to see whether this pathway, this heterochronic pathway, which is still present post-embryonic development and into the phases of synapse formation, plays a role in the maturation, the further maturation of these neurons and their synaptogenesis. And so that's what we've started out to do. First, we're characterizing normal mouse in vitro, and then moving on to human in vitro, and then ultimately the ES cells and manipulations of the microRNAs. We focused on the bipolar cells because the bipolar circuit is very well described. We can both look at the ribbon synapse formation using EM. We can record from both the pre- and post-synaptic sides of the bipolar cells, as well as their interaction with the amacrine cells. So because the circuit is so well defined, we can see how well does it develop normally, how well does it develop in the ES cell-derived cultures, and then how well, whether it's accelerated with the microRNAs. And I should just give you a little bit of data now on what we've done in the past year or so. We've begun to look at normal mouse development in vitro. This is an example of the synapses forming in the plexiform layer of the bipolar cells and the amacrine cells. This is the density of normal horizontal cells and the density of rod bipolar cells. And we see in vitro cultures now, when we start really applying quantitative analysis to see what level we can get in vitro development, we see very good horizontal cell production and differentiation, good bipolar cell production. None of these cells were all generated in the culture, but they array appropriately. They form a good plexiform layer. They're thinner, but they do form the sublaminae within the plexiform layer, inner and outer plexiform layer. Their arbors are slightly smaller, well, probably 50% smaller than a normal retina, but we lack the stretch of the normal retina that we have in culture. Nevertheless, a lot of these same developmental processes go on in vitro. We've also started doing an analysis of the human fetal retinal development in collaboration with Anand Swarup at the NIH. There hadn't really been a comprehensive RNA-seq analysis of the developing fetal retina, so we had to do that in order to track the time of development over time. So we've gone from very early stages, like 59 days, to even much later stages, now 136 days, and we have a comprehensive RNA-seq profile now that we're about to send off. And one of the things we found already, just by looking at culturing the human retina, is pairs of say, 2 pairs of 59 days, one that we acutely dissected and one that we let go in culture for 3 weeks. We find that development progresses in vitro just fine in the human cultures as well. And so, for example, 59 days plus 21 would put it somewhere close to 80 days. And this is the 105-day in vivo. This is the 59 in vivo. This is the 59 plus 21. And so you can see for these rod genes, they're developing normally for these photoreceptor genes. And similarly with the other ages, we can keep tracking. So we can take a 134-day-old human retina and then culture it for 21 days, and it continues to develop. So I think both in the terms of these explant cultures of the fetal retina, we find that their development is proceeding just fine in vitro. So next, we're moving on to look at the embryonic stem cell cultures. We're looking at the retinal organoids and using EM to look for the evidence of synaptogenesis. Rachel Wong has a technique called dialystics, where she can take small beads covered with dye and then essentially blast these little organoids, and then we can see the morphologies of their cells and see whether they're adopting normal morphologies. This is an example, or here's an example of a ganglion cell with a little axon going out. And so by doing that, we're characterizing the size of the dendritic arbors, the ganglion cell subtypes, and the cells that they make synapses with. So I think we'll be able to have a full description of the degree to which the circuit normally forms in organoids. Fred Rieke then can patch onto these cells, record from them, and find cells that resemble ganglion cells and amacrine cells sitting at the top layer of the organoid culture. So again, these things seem to be developing appropriately and forming appropriate synapses, although they're a little bit slower than the normal retina would be when they're ganglion cell derived from an organoid. And then lastly, we plan to manipulate the microRNAs to try and see whether we can speed this process up. This just shows the human fetal retina samples that were developing these are just in vivo prepared samples, but you can see the microRNAs for this heterochronic pathway are generally going up in the human samples as well. It's also interesting, the fovea we find, which is a region of specialization within the human retina, actually develops in a little bit of an accelerated fashion compared to the rest of the retina. And we find that these heterochronic pathway microRNAs increase more rapidly within the fovea. So our goal then is to actually put elements of the heterochronic pathway back into the retinal organoids and then see if that speeds up their circuit maturation. I should say, in reference to to Steve's talk, when we talked at the last Allen meeting and we heard him talk, I thought, well, it would be nice to have an independent measure of time in the retina, because what we have is we can measure the developmental time based on the RNA-seq, the presence of these different cell types, et cetera. But if we really start to manipulate the time in the retina, it would be great to have something that was an independent clock. And so that's when we turned to him and sent him the same samples that we used for doing the RNA-seq analysis. We took the DNA and did the Illumina methylation array, and then sent the data to Steve, and then he presented that. And what's interesting about that, this is the data from plotted again. What's interesting about the having this correlation now is that it provides a way in which we can independently measure the age of these embryonic stem cell-derived organoids to see whether they're tracking at the methylation age with the human fetal retinal samples that we have. And I think that's going to turn out to be pretty interesting. I don't know that methylation is necessarily the driver, and I really have no idea how you would connect methylation to the microRNA pathway, but I think having an independent measure of time in culture or of time developmentally will actually be a really nice benefit and adjunct to our ability to track the normal RNA-seq data and developmental pathway. So with that, I'd just like to close. I'd like to thank Fred and Rachel, who couldn't be here today, as wonderful collaborators on this in our ADI team, Akina and Chi and Olivia Bates, who've all been instrumental in doing some of the work that I described, and then Steve and Ian Glass, who is our source of human fetal retinal samples. Thanks.
We have time for one quick question, if there is one.
Yeah.
So I didn't quite get in the first part. When you knocked out dicer, then the progenitors produced only ganglion cells.
That's right.
But did they stop at the expected time, or did they continue
They just
producing
kept going.
in perpetuity?
Well, up until probably, so we looked only about 1 and 2 weeks after birth. Because what happens is they keep producing ganglion cells, but the ganglion cells have nowhere to go, and they don't make anything else. So this is long past when neurogenesis would normally end.
Okay.
It just keeps going.
Okay.
But eventually the ganglion cells die because they have nowhere to
Right.
go, and then we sort of, that part of the retina where we knock dicer out becomes kind of a rudiment.
Because that's important to say it's an effect on the clock as
Right.
opposed to transformation of cell type.
Exactly.
Yeah.
Well, and additional evidence for that is that the progenitor cells themselves can be distinguished from this early state where they're competent to generate the early cell types and the late state where they're competent to generate the late cell types. And those markers, those molecular markers, the SOX9 and the ASCL1, we see those change as well. So that's the key point. And I think this is the only perturbation over, I don't know how many years that people have tried to understand this clock. This is the only perturbation that actually changes those markers in the progenitor cells. So I think that there are many things that will affect the cell fate of the progenitor cells, but the dicer seems to be the only thing that can stop the clock.